Hello everyone. Today we will discuss about different methods of cell lysis. So it depends upon the type of cell that you want to lyse, what methods can be used. So some cells they have cell wall and they are difficult to lyse, and some cells like animal cells they lack cell wall. So by simple methods also you can lyse those cells. So first you consider what type of cells you will have to lyse, and then accordingly you can select a method. So there are certain methods called as physical methods, certain methods they are called as chemical methods, and there are certain methods which are biological or enzymatic methods. So physical methods uses some physical forces to lyse the cells. So in that under this category the methods include you can use pestle and mortar, and you can with the help of mechanical force applied with the help of mortar you can crush the cells along with the abrasives. Abrasives are used to damage the cells. Instruments like French press can be used, by which you can apply a pressure on the cells. You can apply negative as well as positive pressure on the same cell by trapping it in a small hole called as orifice, and because of this change in pressure the cells can get lysed. You can use blenders to lyse the cells, or you can use homogenizers which uses thick glass tubes and a shaft which is having very uh, less difference between the diameter of the tube and the diameter of the shaft. So cells they get trapped between this space between the shaft and the wall of the tube, and when the shaft moves with a high speed, because of that mechanical force cells can be lysed. In case of physical methods, one different technique, that is ultrasonication, can also be used. Sound waves with very high frequency are produced, and these waves can damage the cells and can lead to lysis of the cells. In case of chemical methods, you can use solvents, different types of detergents. Solvents they affect the structure of the membrane. The lipid bilayer which is present that get affected, the proteins can be denatured, and because of the damage in the cell membrane the cells can be lysed. Detergents also have hydrophilic and hydrophobic properties, and that's why they damage the cell membranes, and when the cell membranes are damaged cells may be lysed. Alkali or acids can also be used to lyse the cells, but majority of the times they can affect the molecules that you want to purify or separate. That's why these are not very preferred method for cell lysis. In case of enzymatic methods, depending upon the type of cells and the cell walls present, you need to select a particular enzyme. If you want to lyse the bacterial cells, then lysozyme enzyme is used, which break down the bond between N-acetyl glucosamine and N-acetyl muramic acid present in bacterial cell wall. And when this bond is damaged, the cell wall structure gets affected, and changes in the osmotic pressure leads to osmotic shocks to the cells, bacterial cells, and then cells can be lysed. In case of plants, the cell wall is made up of cellulose, and that's why cellulase enzyme which will affect the cell wall made up of cellulose can be used. Animal cells they don't have cell wall, and that's why by just changing the osmotic pressure of the cell you can lyse the animal cells. In case of fungal cells, the cell wall is made up of chitin, and that's why chitinase enzyme can be used to lyse the fungal cells. This is a short overview about different methods which can be used to lyse the cells. Thank you all.